Good morning, everybody. I'm Renisha Karki, a fourth-year PhD student at Raskin Group in Department of Civil and Environmental Engineering. And it is my great honor today to introduce our first speaker, Professor Chris Lawson. Professor Lawson is an assistant professor at Chemical Engineering and Applied Chemistry Department at University of Toronto. He's the PI in Microbiome Engineering Lab and also Biozone. It's a Center for Applied Bioscience and Bioengineering. Professor Lawson received his bachelor's and master's degrees from University of British Columbia. He then obtained his doctoral degree from University of Wisconsin-Madison and continued his postdoctoral research at Berkeley Lab. His research is centered on several key areas, such as developing quantitative aspects of systems-level understanding of microbial metabolism, designing and optimizing engineered microbial consortia, and advancing the recovery of renewable bioenergy and bioproducts from waste streams. So please join me in welcoming Professor Lawson.
Okay, great. Thank you so much, Renisha, for that wonderful introduction. And thanks to Betsy and Anne and the organizers for having me out here. We had a really wonderful talk yesterday in the workshop, and I learned a lot about many different areas of the microbiome and how they're connected. And I'm looking forward to hearing more from the other speakers and from you all here today. So today I'm going to be telling you about some of the work our lab does on retooling microbiome engineering for converting waste carbon into valuable products. And so, see how this works here. There we go. So of course, in society, right, we generate a lot of different waste streams. Globally, we generate about 5 billion tons of municipal solid waste a year. So it's a really big number. About 1 billion tons of that is organic in nature. And so in my life, I like to think about how we might use these different buckets of waste. Some of them are more wet in nature, food waste, especially when combined with paper waste. Those things are more wet in nature. Some of them are more dry in nature, so wood waste, plastic waste. And so what I think a lot about is how we might convert all these different carbon sources and use them as feedstocks for making chemicals. And so can we replace chemical manufacturing with, instead of using oil as a feedstock for making chemicals, can we use waste products, right? And so when we think about this, there's two really key technologies that have really reached commercial scale applications. And so the first one is called gas fermentation. And so in this technology, generally speaking, these more dry fractions of organic waste are converted to what's called a syngas. And so this is a mixture of carbon monoxide, hydrogen, and CO2 that is just then fed to anaerobic microbes, gas-eating microbes that are really efficient at converting electrons in those feedstocks to products. And so, of course, you can also directly feed different waste gases emissions, say from steel mills that are also generating syngases, to these microbes to make fuels and chemicals. And so that's how we're generally dealing with the dry feedstocks currently. On the wet side, we use a technology called anaerobic digestion, which has been around for over a century. And so this technology is very efficient at breaking down food waste, some paper wastes, things like wastewater biosolids, and converting those into chemicals and fuels. And the main outcome of this is really to prevent these sources going into the landfill. So of course, if we don't divert this waste and we send it to a landfill or somewhere where it just stays, ultimately it ends up getting decomposed in the landfill and resulting in methane emissions. And so a lot of my work is focused on thinking about how we might retool some of these technologies to really make production of fuels and chemicals more widespread and more economically feasible. And so to start in my lab, we've really focused on that first technology, anaerobic digestion. As I mentioned, anaerobic digestion has been around for over a century, and one of the beautiful things about anaerobic digestion is it has kind of a wide mouth, right? So it can take in a wide variety of different feedstocks, food waste, paper waste, biosolids, as I mentioned, and it can break those things down and produce biogas. So biogas right now is generally the main end product of anaerobic digestion. And what our lab is thinking about doing is how might we make products using anaerobic digestion that go beyond biogas. So like I said, AD has a wide mouth, but it kind of has a narrow spout. So right now it can only really make this biogas, which is great, but could we make other fuel and chemical molecules using this technology? And so when we ask that question, oftentimes we're confronted with, the biology is too complicated. We don't have a lot of tools for being able to do this type of manipulation. And really, there's these empirical ad hoc methods to drive the technology. And so what my lab has really been focused on is trying to open up kind of this black box. We call it sludge, so that just indicates how complicated this technology really has been. We try to open up the sludge, if you will, and study the microbiome, and more importantly, develop new tools to manipulate its function. And so key questions in the lab that we're asking are, can we better understand the microbes and the functionality of those microbes in these processes? Can we more precisely control the way those communities are assembled and how they function? And then can we manipulate, maybe using genetic approaches, the biology in these systems to make non-natural products? So many of the chemical products in society we're trying to make, for example, one can't make just via natural selection or the native pathways that are present in these organisms. And so really this requires kind of the merging of more traditional environmental biotechnology approaches with advances in metabolic engineering. Right, and so to address these questions, really we need new tools to both understand and manipulate the biology of these systems. And so as an overall vision for my lab, this is kind of the approach that we've been trying to pioneer, which is this top-down, bottom-up approach for developing synthetic consortia that are able to be used as a platform for converting all those complex organic feedstocks into chemicals and fuels. And so the way this vision works is we first have an initial microbiome. Maybe it's from the cow rumen, or maybe it's from an existing anaerobic digester, or a lot of the microbes present in those systems have the functionalities that we want to work with or that we want to engineer, but aren't perfectly fit yet. And so what we do is we take those microbes, we inoculate them in a bioreactor, and we use selection as a tool to manipulate the function of those communities. And so maybe we'll drop the pH, we'll change the loading rate or the retention time in that reactor, and we'll select for the functions that we want to achieve. And so with that, using selection, that's been an extremely powerful approach and successful approach in environmental biotechnology. But sometimes it doesn't get you all the way there, especially if you now want to make non-natural products. And so what we've been really focused on is from those systems, can we do high throughput isolations and isolate, and biobank a large amount of the diversity, the functional diversity from those communities, and split them into what we call different functional guilds or functional groups of microbes. And so those organisms, for example, that might be taking arabinoxylans in the feedstock to lactate, or lactate to a different product, okay? And then we can characterize them, their physiology more systematically. And then what we do is, with the microbes that we like, we can either rationally combine them into synthetic consortia that make the products that we want, or what we've been really focused on is applying what we call this design, build, test, learn cycle, where we're mixing and matching different microbes and we're screening them and using machine learning to drive the systematic design of these consortia for a particular application, in our case, fuels and chemicals. So I'll talk kind of about the different tracks in the lab. So I've been at University of Toronto now for 3 years, and we've been focused on, several of these tracks, and now we're finally in a place that we're starting to bring it together. And so throughout the talk, we'll hit on a number of these different technologies. The last thing I should mention is that, of course, once we can isolate these microbes, is we can actually also start to develop genetic tools for them. And so we can manipulate their metabolism and then also reintroduce those into the synthetic microbiomes that we're developing. And so the main questions were, why are we doing this, right? So why is this approach powerful? And so we think that by using this approach, we can boost what are called the titer rate and yield metrics, these so-called tri-metrics of bioprocesses that are largely some of the key parameters driving industrial biotechnology approaches. We think that these more reduced synthetic communities are extremely useful experimental systems for better elucidation of the underlying microbial interactions and functionalities in these systems. If you study just the sludge itself, it can be really challenging to actually untangle what's going on. These more reduced systems offer an experimental platform to be able to actually elucidate mechanism. And of course, can we use these systems to make higher value products, as I've mentioned. Okay. And so, thinking about higher value products, what are the kind of products one wants to make from waste feedstocks? So as I mentioned, focusing on anaerobic digestion, the technology so far has been very good at making products on the bottom of this so-called, what we say is a value volume pyramid. So you can imagine in society, we can make fuel products, and those products, we need a lot of those products in society, right? They have a very, very big volume, but their market value is not very high. At the top end of that pyramid, we could go make something like a pharmaceutical molecule, a fine chemical, very high selling price, but the volume that we need, right, if you think about different injections, very, very small. So if we're focusing on climate change as a problem, we kind of want to be either at the bottom of this pyramid or somewhere in the middle in the bulk chemicals and plastic space. And so as I mentioned right now, anaerobic digestion can make things like renewable natural gas, which is great, but could we also use the technology to kind of move up this value volume pyramid to the bulk chemicals and polymer space? And so from doing so, we can make higher value products that might drive the economics of it, but there's a couple other reasons that might be valuable for doing so. Okay, and so, yeah, what products do we make when we look at opportunities for offsetting greenhouse gas emissions? Certainly the chemicals and petrochemical sector is a very large target for molecules that we want to be making, but also these long-haul transport fuels. So you've probably heard of sustainable aviation fuels, fuels for boats and ships. These are things that inherently are very challenging to electrify, right? And so could we use biology and these waste feedstocks for making some of those products as well? And so one of the other last things I want to mention in kind of this introduction to the topic is what is the waste, what does the refinery of the future look like? So this was a really nice article that recently came out in Nature talking about kind of the waste or the refinery of the future, right? So in today's world, largely our feedstocks are oil, right? And other products, largely this other is natural gas. And we're using, chemical physical methods largely to make things like refine that oil and those products into transportation fuels, and then things like polymers and chemicals. And so in the future, it's anticipated that this is really largely going to be changing. One, driven by the fact that things like transportation fuels are largely going to be electrifying. And so our products then are again going to be kind of some of those long haul transport fuels, jet fuels, diesel for boats, and then polymers and chemicals. And the question is, can we replace oil and natural gas as a feedstock with plastic waste or municipal solid waste, biomass, and then of course CO2. And so this is really in line with using CO2, agricultural, and municipal waste as feedstocks for chemical manufacturing. And so just to underline this further, right, the future of the energy sector is largely going to electrify. So these are just some stats looking at how the energy sector might transition, again, away from carbon-based sources of energy, more to non-carbon-based sources of energy. And that makes sense, right? We don't need carbon as a feedstock for generating energy. We can use solar, wind, hydro, those other kind of things. And so this really further emphasizes focusing our technologies of the future for dealing with waste that inherently has a high amount of carbon to make products that need carbon in them, right? So these chemical and polymers of the future are carbon-based products. So these are things that we're going to need. Okay. And so as a starting point, our lab is focused on a product called medium chain fatty acids and the various family of medium chain oleochemicals, their derivatives that come from them. And so what are medium chain fatty acids? They are C6 to C12 monocarboxylic acids. And so you've heard of short chain fatty acids, of course, acetate all the way to valerate. The medium chain fatty acids start from the C6 and go up to the C12. And naturally, microbes can make the C6 to C8 variety. And so they're a useful starting point for retooling these anaerobic digestion processes away from biogas toward chemicals because they're more valuable than methane, so they have a higher selling price that could drive the economics. They're using a wide variety of products, so you'll find them in nutraceuticals. I went to the local Shoppers Drug Mart, it's a Canadian store in Canada, and you'll find it in personal care products, different industrial chemicals, and one of the other big markets is just as a animal feed additive. So they're also used in animal feed. So they have a wide variety of different applications. They have a higher selling price. The two sources currently for making MCFAs are from oil refining, but also a big portion come from palm and coconut tree refining as well. And so obviously that's not great if you've read anything about palm and coconut oil refining because those plants are leading to large deforestation activities. And so if we can make processes that can make these MCFAs more sustainably from a local source, obviously that would be another big environmental benefit of making these MCFAs. And finally, MCFAs themselves can be derivatized, used themselves obviously, so they're valuable, but they can be made into a whole suite of different derivative products that also have their own large market sizes and different applications. And so really, if we can retool the biology, the microbiology of these digesters to not just make biogas, but make a whole suite of these products, it would really enable kind of the so-called biorefinery approach, putting biotechnology at kind of the heart of that refinery of the future that I mentioned on the couple slides previously. Okay, so how do we actually turn waste products into medium chain fatty acids? And so, one of the main starting applications in our lab has been thinking about taking food waste, here's green bin waste, and turning it into these medium chain fatty acids. And so the way this works, if you're familiar with kind of the classic anaerobic food web, right, is in this waste, we have a lot of complex polymers coming in, cellulose, hemicellulose, starches, peptides, lipids, and ultimately, if we let those things sit and break down all the way, and you'll get complete decomposition to a reduced product, methane, and then an oxidized product, CO2. That's the way kind of anaerobic fermentations work, right? And so we have different steps. We have the hydrolysis of these more complex polymers to their monomers, and then subsequent fermentation to things like short chain fatty acids, lactate, and ethanol, going all the way to methane. And so in the lab, what we can do is we can do things like drop the pH of these systems just in a reactor. We can inhibit methanogenesis. And then what we select for is a process called chain elongation, where we have a lot of these complex polymers funneled through to two key intermediates, ethanol and lactate. And that ethanol and lactate are key substrates for a group of microbes called chain elongating bacteria that take the ethanol and lactate to these medium chain fatty acids using a pathway called the reverse beta oxidation pathway. So two really key groups of microbes, there's many microbes in these systems, but two key groups or guilds, if you will, are the lactic acid bacteria that can break down these complex polymers, making lactate, and then these chain elongators that can take the lactate to MCFAs. So this is the so-called lactate-based chain elongation process. And so this is a great technology, but when we just use selection, of course, there's some limitations of the process. And so with any digestion process, hydrolysis, how much complete breakdown of those complex polymers you get can always be a challenge. Selectivity of the product, so how much of that carbon feedstock do we actually convert to our MCFAs, which really is really key for determining the economic feasibility of the process, is a very key metric and goal for an engineered system. Currently, if we use a technology like this, the product spectrum is still relatively limited, right? So we can make C6 and C8 products. We can't make the C10 and C12 other MCFAs that we're interested in making. We can't make all those derivative products. And so it's still you end up not quite this refinery of the future that we're going for. And probably one of the biggest things that I'll comment on at the very end of my talk is that we lose half of the carbon. So whenever you do polysaccharide-based fermentation processes, you've heard of bioethanol, whatever processes you're running, this is no different. Half the mass of carbon goes up in the air as CO2. So if you're in the business of converting a carbon-based feedstock into a chemical and half of your feedstock's going to the air as CO2, obviously that's maybe not a great business model, right? And so these are all some of the limitations with the current technologies, state-of-the-art technologies that we're hoping to address with synthetic communities. And so kind of like as a vision, what we'd like to do is build these synthetic communities that address some of these bottlenecks or targets of the existing technology. And so the way this would work is we would have our complex polysaccharides here, or polymers, an example here being starch. We have this group of microbes, the lactic acid bacteria, that will break that down and ferment it to lactate. And then we have our chain elongators that would take the lactate and make the MCFAs. But of course, in the process, what happens is they emit CO2. And so then can we add renewable sources of energy, hydrogen from water electrolysis, syngas from gas fermentation and gasification, as I mentioned earlier in my slides, to really drive or feed these other group of microbes called the cetogens that can recycle the CO2 with those reduced electrons back into products to make chain elongate MCFAs. So this is kind of the blueprint that the lab is broadly working on. And of course, in 3 years, we haven't done all of this yet. But what we started with is chain elongators, so focusing really in detail on the chain elongators. And so I'll start the rest of my talk, describing some of the work we've done on chain elongators, and then show some of the work we've been doing to build out synthetic communities from these groups of organisms. Okay, so if you're in the business of making synthetic microbiomes, you have a lot of decisions to make, right? Here's a list of chain elongating microbes that we currently have in isolation. The first description of these chain elongating microbes actually is back to the 1930s, and subsequently thereafter, other microbes like Clostridium clavari, kind of a famous chain elongating microbe, was discovered in the early 40s. And really with the advent of,
That's the original size in the pin. So let's go down here, the little up arrow.
Could you mute yourself online, please? Oh, that's okay.
And oh, great! You're already in.
Sweet. Yeah. Yeah. All good. Cool. They were really keen on the chain elongators. Yeah, so we have this, this long list of chain elongating microbes that really has been taking off. The number of these chain elongating organisms that have come into isolation, really with the development of these mixed culture technologies. There's been a lot more attention paid to chain elongating microbes. And so you can see what these organisms are not necessarily taxonomically unified, right? And so you see some come from the Megasphaera, some come from Clostridium, some are coming from Pseudoramobacter. So their functionality does not necessarily correlate with their taxonomy, so that's an important piece. And they also come from quite different environments, right? And so this is from the sheep rumen, this is from a bioreactor. My favorite microbe, as you'll see why soon, is coming from a tooth infection, right? So lots of fun exotic sites, right? And so when we're looking at these synthetic communities, starting with the chain elongators, there's lots of questions to ask. So the first one is, which chain elongator are we going to use in our synthetic community, right? There's lots of them. Which one do we pick? What are the differences between these chain elongators? What are their phenotypes? What are their ecological niches? What are their differences between each other? Can they be genetically modified? As I mentioned, these organisms kind of sit at the business end of our process. If we can't genetically modify them, it's going to make it difficult to work and scale up the process. And do they form stable synthetic communities, which is a really important question, obviously, if you're looking to make a synthetic consortia. Okay, so lots of questions, only so many grad students. And so what we did when we started the lab is we really invested in automation technology. And so, and we wanted high throughput approaches to really be able to screen the physiology of these organisms and develop genetic tools in synthetic consortia. So if you want to do anaerobic microbiology in high throughput, the first thing you kind of need is a very large anaerobic chamber. And so that's what we have built here in our lab. And then the other thing you're going to need is some automation equipment. And so we've then put a number of different automation equipments inside of our anaerobic chamber, things like liquid handling robots, incubators, plate readers, and shortly we'll be getting things like colony pickers as well. And having these systems integrated robotically such that we can develop high throughput experimental workflows to really be able to address these questions and nail and play catch up with these microbes. Because many of them, unlike E. coli, yeast, or maybe some of the other model organisms you work with, these organisms don't even have a growth curve, never mind kind of genetic tools, right? Okay. And so here's kind of the initial system that we had in the lab. It's been expanded a little bit, but the core of it still so far is a liquid handling robot, incubator systems, and a plate reader. And then we have, that connected to things like GCMSs and HPLCs. And really what this allows is high throughput screening of thousands of different microbes at a time. And so this is really efficient because the traditional approach is usually, tens of Hungate tubes that we're screening or one bioreactor per PhD student, which, is a really low throughput kind of approach. Okay. And so we've come back to those questions, what are the physiological features of these chain elongators? Which ones should we use? And so we've been trying to address those questions with our robot. And so what we've been doing, I'm just showing a subset of the chain elongators that we've been screening. What we can do is we can grow these in high throughput, and we can look at their growth on different substrates. For example, here I'm just showing 3 sugars and lactate, and we can see how they grow over time and how well they grow on those substrates. And we can also look at their product spectrums, right? So we can see, are they making the MCFA products that we want? Are they making butyrate? What other products are they making using the platform? And this allows us to get key stoichiometric and kinetic parameters that then inform what microbe we might be choosing when we decide to make our synthetic community. And so here, one of the organisms that stood out in our initial screen was this tooth infection chain elongator, Pseudoramobacter lactolyticus. And notably from lactate, it really makes quite high specificity of these C6 and C8 MCFAs. And so this was an exciting microbe, and we've started to screen all these different banks of chain elongators. We can also screen, of course, many other substrates. This is still quite not a long list, but a more exhaustive list looking at the different maximum growth rates that these organisms make. And here I'm showing you red, the different concentration of products of medium chain fatty acids from C6 to C8, but also some of the shorter chain fatty acids, C3 to C5 here. And one of the nice things here that was really interesting is it's allowed us to identify other substrates that are not, naturally described for these organisms that they're good at also making MCFAs with. For lactolyticus, one of them was also glycerol. As I'll show later on, we've also found some microbes that are really efficient in using things like amino acids to make MCFAs. So really exciting, showing that the versatility of these microbes is beyond what might already be currently described in the literature. One of the other things that we've looked at, so when we're trying to make a microbiome, we want to pick microbes that grow the fastest, that make the most product, but there's other phenotypic traits we're also interested in, of course. So things like environmental tolerances, pH to pH, but also the products. So MCFAs themselves are actually quite toxic. And so here's kind of just some of the screening we've done looking at the toxicity and the tolerance of MCFAs on these chain elongating microbes. And so in blue, we have some of these, more model organisms like E. coli, which have actually been genetically modified to make MCFAs. It's a different approach. And what you can see is that things like E. coli are pretty crummy at actually tolerating medium chain fatty acids, especially relative to these gram-positive chain elongating microbes that are naturally occurring, like a lactolyticus. And so you can see the chain elongators that are native, don't really get affected by C6 concentrations too much until they're very, very high. But then when we get to octanoic acid, the toxicity obviously is a lot higher, but still these gram-positive chain elongators are still quite robust. And we've also started to screen things like decanoic acid, so C10, which again is a product that they don't tend to make. And so what you can see here is decanoic acid, especially if you look at the scales on the x-axis here, is extremely toxic, even to the chain elongators. And so that might point to a mechanism as to why chain elongation naturally actually doesn't go beyond C8, which is something we've been looking at as a genetically engineering target. And of course, this tooth decay microbe, my favorite, lactolyticus, is again really tolerant for high concentrations of these medium chain fatty acids, including some concentrations of decanoic acid too. Okay, and so we can look at environmental tolerances. We can look at just classic stoichiometric and kinetic parameters. One of the other things we've really investigated too are different selection parameters that drive longer chain length product formation. And so here, this is a really interesting study that was initiated by my PhD student Ian Goyce, where he was growing a lactolyticus under different ratios of lactate to acetate, okay? And so what he observed is that when you have higher concentrations of acetate in the media, the chain elongators grow, the lactate-based chain elongators grow a lot faster. But interestingly, when we looked at the acetate concentrations, they didn't really consume them. So acetate was in the media, but it wasn't really being consumed very much. And so the other interesting observation with this is that as they grow, or as you vary their lactate to acetate ratio, the product spectrum shifts from more short chain fatty acids, in this case butyrate primarily, and some C6, toward the longer chain products, right? So when they grow slower and they have a higher lactate to acetate ratio, they get longer chain products, which from a biotech perspective is what we're interested in, but they grow slower. And so this is a really interesting kind of question, like what explains these trade-offs? And I love these kind of questions, like they really point at the heart of kind of resource allocation and kind of some core ecological questions that are tied to the metabolism of the microbe, right? And so we've been trying to explain this trade-off for quite some time, and I'll be brief with this explanation of what we think is happening here, and of course we can chat about it more at the break. But what effectively we think is happening is this difference in phenotypic capabilities between the ability to recycle acetate versus assimilate acetate. And so let me explain this here. And so if you're in the business of making medium chain fatty acids or even say butyrate, what you can do is first you use lactate. Let's just say we're starting with lactate as an electron donor. What is used is the organisms will oxidize lactate, right? Generating reducing equivalents to make acetyl-CoA. And so if they're assimilating acetate, what they can do is they can bring acetate into the cell and also bring it to their acetyl-CoA pool. And so now you have 1 mole of lactate and you have 1 mole of acetate and 2 moles of acetyl-CoA. You can combine those 2 moles of acetyl-CoA and run this reverse beta oxidation cycle, which is what's generating the medium chain fatty acids, and you subsequently reduce those 2 molecules of acetyl-CoA to make butyrate. So if you go around the cycle once, you're making butyrate. If you go around the cycle twice, you're making C6. The medium chain fatty acid, 3 times, you're making C8. And so when these organisms take 1 mole of lactate and 1 mole of acetate, and they combine them, if we do our redox balancing, so bringing you back to kind of first year biochemistry here when you're asked to account for redox, when you oxidize 1 mole of lactate, you're generating 2 electron equivalents of NADH. And when you balance it, the pathway requires 3 moles of NADH, but you're also generating 1 mole of reduced ferredoxin. So if you look in the red, you have 2 moles of NADH, 1 mole of reduced ferredoxin. So you have 3 reducing equivalents that ultimately can be balanced with these 3 electronic donors that are required to run the RBO cycle, right? And what happens here is because of this imbalance between ferredoxin and NADH, they utilize this enzyme called the RNF complex to take that ferredoxin and oxidize it, transfer electrons to NADH. And because that process is thermodynamically favorable, they can actually pump some protons, and this is one of the main ways they generate energy. So when they're taking acetate and they're assimilating it with 1 mole of lactate to make butyrate, redox is balanced, okay? So the problem is, what happens when acetate starts to fall down? When you get limiting acetate, right? Which is that trend I showed you when the acetate's available, they tend to make largely butyrate. So they go around the cycle once making butyrate, largely because they can assimilate the acetate and also bring in 1 mole of lactate. When acetate's not available, what you have now is you have to oxidize 2 moles of lactate, right? Because now you have to still generate 2 moles of acetyl-CoA to run the cycle, and so you have 4 reducing equivalents of NADH to balance plus 1 ferredoxin. And so the redox doesn't balance, right? And what you'd have to do to actually balance this is you'd have to run this RNF complex in reverse, which isn't thermodynamically possible. So you can't do it. You actually can't, on 2 moles of lactate, thermodynamically make butyrate because the redox doesn't balance. And so what the organisms have to do instead, because of this redox constraint, is actually go around the cycle twice and pull in 3 moles of lactate to run the cycle. And you can see here with 6 moles of NADH, 3 moles times 2 going around the cycle twice, you get redox balance and you can burn that ferredoxin as hydrogen and that can leave the cell. And so this is some interesting modeling work that we've done to show this and explain this. And on the next graph here, we kind of just summarize this trend where as an organism makes longer chain products, they have to ultimately, they end up recycling acetate more than assimilating it. And so this is really to essentially mitigate redox. And so this explains really why acetate recycling is required in these organisms to make these longer chain MCFAs, and really points to their one of their main ecological niches. And then the other thing that we wanted to explain is, of course, why does the growth rate of this organism dramatically fall off? So we've explained why their product spectrum pushes to C6, but why does their growth rate come down? And so one of my other students, Connor Bowers, who's done a lot of really interesting resource allocation modeling, what he's done is he's looked at thermodynamic models to estimate enzyme costs to carry a certain amount of flux for a pathway. And what the calculations show is that in order to make, when you go around the cycle once, you ultimately need less protein in the cell. So you can imagine a cell has 10 proteins. If you have to push more of those proteins toward catabolism to drive the fermentation, there's less proteins available to actually grow. So the growth rate comes down, right? And so in this case, you actually, to make C6 go around the cycle multiple times, it requires a higher fraction of your proteome allocation toward catabolism. And so then the growth rates fall down. And so there are some really nice modeling studies that came out of this, right? And so the bottom line, if you didn't get that, was that acetate recycling requires longer products, but it also increases the catabolic protein burden. And so this is why we see this slower growth. What's even more interesting about this, though, is that in our biobank of microbes, some of them only make butyrate, and they stick to butyrate, and some of them will do this acetate recycling and make longer chain products. And so what we really asked is, what's the benefit of this? Like, what's the ecological reason why a microbe might make these longer chain products? And it really comes down to what we've shown here in this study that we're hopefully submitting soon, is efficient lactate utilization. So you can imagine if you have to use lactate and acetate, right? If your acetate runs out, you can't actually use any more of the lactate if you can't actually do this recycling mechanism. And so a lot of microbes in the human gut, for example, Lactobacillus, Acoccaceae, these classic gut microbes that only make butyrate we've been looking at, they don't do this recycling mechanism. So here I'm showing you the chain elongator, Lactolyticus. When under acetate limiting and abundance conditions, it maintains its growth, it can fully grow up, and it tends to make longer chain products. In contrast, organisms that are sole butyrate producers, they die off. So right when their acetate is depleted or becomes limiting, they stop growing, and they can't actually consume all of the lactate that's available. And so this really points to the ecological niche of these chain elongating microbes and where they're most fit. And it's really then, in our opinion, that chain elongation towards C6 and C8 fatty acid products is really a function of, or an optimization of lactate utilization capabilities. So this was kind of a really interesting study. Again, that from screening, we were able to pare down and start to drill down into more mechanisms. Yeah. Okay. One of the other quick things I'll mention is that we've also been developing genetic tools for these microbes. I won't belabor this point too much, but one of the issues when you're trying to transfer plasmids into these anaerobic chain elongating non-model microbes is they have restriction modification systems. And so those are essentially native immunity systems that if a foreign DNA is not methylated correctly, the plasmid's going to get chewed up and spit out, and you can't transform the microbe. And so what we've been working on is a methylation pipeline where we take the organism of interest, we learn its methylation pattern, its native methylation pattern from methylome sequencing, and then we build an E. coli strain in the lab that expresses the correct methylase to methylate the DNA. And then we've been doing conjugations and transformations with that. And so this is just an example of this where we've taken Clivery. You can never transform Clivery if you try without this approach, but when we do this, we can now transform Clostridium Clivery, one of the classic chain elongating microbes. And so this really opens up the opportunity for being able to do a lot of the metabolic engineering goals that we are interested in pursuing in our lab. Okay, I'm sure I'm a little bit over time, but I'll step it up a little bit. And so we have this screening platform. We've learned a lot about the physiology of these chain elongators. We've developed genetic tools for them, but now the question is, can we really, bring it all together in this, source split recombined kind of approach? And so that's what we've been really focusing on now, especially over the last year. And so to answer that question, I'll bring you to the tipping floor of one of the anaerobic digester facilities in Toronto. So there's two main facilities where all the organic waste goes, Dufferin and Disco. And what happens is you have all the organic waste in garbage bags. It gets separated from the plastic bags, and you generate this nice, yummy pulp. Yummy for the microbes anyways. And at the facility, they're making biogas, but we're trying to turn this pulp itself into MCFAs. And so just some characteristics of the pulp. It's actually really a nice feedstock for making MCFAs natively, because in the pulp itself, it's very high abundance of lactic acid bacteria. So those microbes that break the food waste down to lactate. And so 70% is lactic acid bacteria. And actually in the buffer tank prior to the digester, this is kind of like a short retention tank that feeds the digester. We actually saw quite an enrichment of putative chain elongating microbes in that tank that we used actually to inoculate our reactor. So we've been running this reactor downstairs in our lab for over almost 2 years now. developing this mixed community that can make MCFAs. You can see it's achieving pretty reasonable yields and titers of the MCFA product. And so the question is, can we isolate microbes from this community and stitch them together into synthetic communities, right? And so what we've done here, this is a collaborative project with a colleague of mine, Harris Wang at Columbia University. He has another robot in a chamber that's complementary to ours. He has a colony picking robot. And so essentially what we did is we kind of did this tour de force isolation campaign where we made medias of all different types of different antibiotics, and then actually just the food waste itself, the pulp. As you can see, these are kind of food waste plates. And what we did is we did high throughput isolations of our community. And the way we isolated is we not only picked random colonies, but we used a machine learning guided algorithm that looks at the morphology of the colony, the color, the thickness, and tried to pick as unique of colonies as possible to capture as much diversity, okay? And so I'll tell you a bit about that in a minute. One of the microbes that we found, I have to share this because I just love this microbe so much, was this novel Eubacterium species. It's quite unique, but it's popped up in a lot of mixed community studies that have been doing metagenomics. And so what's unique about this microbe is this is it growing just in this PYG media. It's a rich media that it can consume lactate, so it's a lactate-based chain elongator. And what you can see here is it's highly selective towards C8. So most of its products on this media anyways are going to C8, which is extremely unique for one of these chain elongating microbes. The other thing that was really cool about it, when we isolated it, we could directly put it back in the food waste pulp, and it did its job very, very robustly, right? So it's still consuming all the lactate in the food waste, producing medium chain fatty acids, right? One of the other really cool things about it is it also seems to be a prolific amino acid degrader. And so this is quite a novel phenotype, thinking about how amino acids are actually direct substrates for chain elongation as well. And so here I'm just showing consumption of different types of amino acids. You can see lysine, threonine, serine, for example. And this is work we're still just getting into and looking at all the pathways and how it's regulated. But again, a really cool novel microbe. Obviously, there's lots of amino acids in food waste from all the proteins that come into food waste, right? So really cool microbe. Okay, so anyways, we recovered thousands of different species that included different chain elongating microbes, different lactic acid bacteria, and these were indeed microbes that were actually also present in our original bioreactor. And so what we've been focusing on now is this so-called design, build, test, learn approach, where what we do first is we take these guilds of groups of microbes and we try to classify them into different guilds. And so we have a number of approaches we're trying to do on that. And then ultimately we have some community design where we're taking different guilds in our community and making a mixed consortia. And we're doing this systematically. We're building them with our robot. So the robot's used to mix and match different isolates. We then screen their MCFA production in batch. And then using our design and our results or responses, we can build machine learning models that recognize patterns between those designs and responses. And then an approach called Bayesian optimization, which can actually recommend new combinations of media to test that have an increased chance of improving MCFA production. So this is a recommender, much like Netflix might recommend different movies for you. In this way, we're using a similar algorithm that's recommending microbes that might increase MCFA production, okay? And so we have just some really preliminary data on this, and then I'm done. And so what you can see here is we took our food waste pulp, we sieved it for this study to make it simple. So we're just looking at the soluble fraction, which is still very rich in complex organic matter. And what you can see here is when we just grow these, we took 85 isolates, we grew them separately, and we measured their growth and their MCFA production. So you can see here, some of them are growing, some of them are making MCFAs, one in isolation. And when we build just 3-member communities, you can see dramatic increases in growth, and then obviously in MCFA production as well. And so this is really promising to us, and now we're really investing in this approach to keep going with different iterative cycles. But even these 3-member communities, it turns out, is very promising, of course. And even just with the 3 microbes and our top designs, they're already producing as much MCFAs as this mixed community, a little bit higher actually. So really promising. We're really excited about this to really hammer this home with our robot. The top 3-member community was some lactic acid bacteria, what we think is a chain elongator, and that Eubacterium species that I mentioned. Made 5 gram per liter of MCFAs just on this initial pass, which is really exciting. And now we're really taking this approach with 200 isolates, which we have their genome sequenced, where we can try to really implement this design, build, test, learn cycle with machine learning on our robots. So the last question that hopefully we can discuss as a group today is, will this thing actually be stable when we try to scale it up? So I have a lot of thoughts on this that hopefully we can have a bit of a conversation about. Okay, so summary points here. Hopefully I've convinced you that chain elongation is an exciting emergent technology for making sustainable chemicals from waste. In particular, we think with advances in synthetic microbiomes, this could really be this platform of biorefinery approach for doing this. The other thing I've showed you is this fancy robot, and more importantly, just the opportunity that high throughput phenotyping and screening and workflows have in helping us catch up to characterize the physiology of all these really exciting and weird anaerobic microbes from the environment that we know very, very little about. We've also looked more in detail about mechanisms, for example, on ratios of lactate to acetate and how that might actually help us describe what a chain elongator is and identify its ecological niche. And now we're in the process of further developing genetic tools for these microbes now that we have the ability to actually get DNA to their cell. And yeah, really these synthetic consortia we think again are really a main approach that will help us recycle efficiently carbon into MCFAs. And what we're hoping to do next is drop in these acetogens into these tricultures. And we already have some preliminary data on this that I didn't have time to show today, that is showing that this approach is actually working. So happy to chat more about that. And so with that, hopefully I'm not too over time. I'd like to thank my amazing team at U of T, who are the ones who are actually doing the work. I just get to come here and speak about it. My great collaborators in Toronto that really help out, especially on the computational side, Krishna Mahadavan and Mohammed Musavi, who's a machine learning expert, Professor Harris Wong from Columbia, who helped us with the isolation work, and 2 other collaborators from SBRC in Nottingham who have been helping us get these genetic tools to actually work in anaerobes. So with that, yeah, hope we can have a nice discussion. So.
Thank you for your talk. This was really interesting. I'm curious about the Eubacterium isolate. So you put it back in the pulp and it was still active, but I'm curious, when you guys were doing the original isolation, how active was it in like the starting system? And like, what is the activity compared to other suspected chain elongators?
Yeah, that's a great question, right? So in our main reactor, this Eubacterium species is anywhere between 2 and 8% in our mixed culture reactor. And we can look at its genome to our megs, and it's the right one, right? When we isolated it, it didn't get isolated on the food waste pulp media. It got isolated on this rich PYG media. And so then the question is, when we isolate organisms in our food waste on other medias, the risk is, well, will it go and grow back in the media? And to our pleasant surprise, we found that even though if we isolate microbes on medias that are different from our source condition, they still grow back. So we've looked at this systematically across our biobank, and like 80% of them will come back and still revive in the food waste, which was very fortunate. Otherwise, it would have been a big slog. So I hope that answers your question. Yeah. So it's about 2 to 8% in the microbiome itself. We think some of these lactanospiraceae organisms are also making MCFAs. Most of them actually are making just butyrate though. So we're still sorting some of these things out. Yeah.
And also with that isolate, I'm curious, have you looked into how it responds to like high C8 concentrations? Like,
Like, is it tolerant of high C8 concentration?
Yeah, because it's producing a lot, so.
Well, it must be because it's producing a lot, but we didn't include it on our initial preliminary screen of all the other chain elongators because we didn't have it isolated yet. So yeah, that would be something good to go back and test. And related to that, we don't even actually know how MCFAs get out of the cell. So one of the main things we've been doing is we've been doing some proteomic studies under different concentrations of MCFAs to try to just identify what the transport proteins and transport mechanism for getting MCFAs out of the cell is. And maybe that's an opportunity to also look at toxicity. We think MCFAs themselves are kind of membrane uncouplers. And so that's something else we've been starting to study. Of course, there's many things we want to study, but only so much time. But we'd like to look at the lipidome of some of these organisms and learn how their membrane characteristics, their lipids, might influence their toxicity or tolerance to MCFAs. Yeah. Good question.
Hi, this was a great talk. Quick question. You mentioned wanting to know about whether or not the bioreactors have these stable communities once you put in these microbes.
Yep.
Have you looked at whether or not your mixed communities, if their relative abundances are shifting as the metabolic reactions proceed? Are you getting a change from your starting ratio?
Yeah, that's a really good question. And so one thing I didn't describe in detail here is how this reactor operates. So fermentation processes can go in two classic modes. One is batch, right, where you feed food waste, the reaction proceeds dynamically, and then products are forming, and that's kind of like the plots I showed you. The other mode is continuous. So you're continuously feeding a substrate, and then your products and your consumption rate reaches a steady state, so the products don't change. Of course, in our food, we're feeding real food waste. So in the food waste, it's heterogeneous and it changes batch to batch, and so we do see some fluctuations in products and things like this. And so then the question is the microbiome also dynamic? We don't know yet, but we've ran this reactor for 500 and some days, and we have samples and DNA from every single day. So yeah, we're going back now and we're sequencing the time series to look at community dynamics. Yeah, and of course, with a reactor like this, operated by grad students, which is a ton of work for whoever is doing this, but really fruitful, doesn't always go perfect, and so there's disturbances and upsets. And so we have some really interesting dynamics. One of the most interesting dynamics was actually, there's two plants in Toronto, one's Dufferin and one's Disco, and you'd think they're the same. They have the same waste going to them, but the Dufferin plant had some upsets, and so we couldn't actually get pulp from them, so we had to switch to the Disco plant. And the feed looks the same, but the chemistry was quite different, and the inoculating microbes in them was a little bit different, and so that really changed our process, actually. And so there's some really interesting things we think we're learning about the process by serendipitously having this feed change in us. Yeah, we've learned a lot from it, so, yeah. Particularly competing processes with making MCFAs. Two main ones are propionate production and methane formation.
Well, really great talk, Chris. I heard you talk about 2.5 months ago, and I'm already seeing a lot of new results. So I'm going to ask something more about the future,
yeah,
if you don't mind, because I expect that you make a lot of progress very quickly. So there are two things. One is, what do you think is the limit of making this medium chain fatty acid? Like, is there a limit you think that somehow exists physically or biologically?
Absolutely, yes.
But then what do you, not just you, like the field needs to achieve in order to make this like biorefinery work, right?
Yeah, yeah, that's a really great question, Nina, thank you. So, the question was, what is the limit for these MCFAs, right? And so we can think of technical limits, and then we can think of economic limits, right? So when you're trying to commercialize something at scale, the economics, and just as we talked about sustainability, that's what this is about. Sustainability is you're meeting your environmental goals, your social goals, and your economic goals, or else the process isn't going to run, right? So technically speaking, the economics is, we've done some techno-economic analysis, is really based on the yield of MCFAs that we can get. out of the process. So how much of that feedstock we can convert to MCFAs. So if we can achieve somewhere around 30% MCFA yields from our feedstock, that is economically seems to be viable. And then it drives the cost, but it also helps us meet our solid reduction goal. Because at the plants, one of the main goals is not just making a product, their primary goal is getting rid of the waste, right? So if you're not making MCFAs at a high enough yield, right? So when you have waste coming in, it can go to the MCFAs, it can go to the gas produced, CO2 and the hydrogen, plus the other stuff. If you can't get a high enough gas and MCFA production, you can't treat the waste, so that's a problem. So that's more of the economics. On the technical side, one of the main challenges, obviously, that this plot shows, which I think is the biggest one, sorry for all the jumping here, too many animations, geez, is this. So MCFAs are really toxic. If we want to make C10 to C12 fatty acids, we've shown in another paper, technically from like a thermodynamic perspective, very feasible, should work, but it seems like they're even more toxic than the C6 and C8. And so I think to deal with this is there's bioreactor innovations, I think, that can happen. So not microbially. So you can extract in situ these MCFAs as they're getting produced to keep the concentrations low. And that's been demonstrated. So that's a key bioprocess innovation that can help the microbes do their job. So if they're making it and we're extracting it right as they make it, that can help deal with some of the toxicity challenges. Hope that addresses some of the questions. But in theory, I think we can get extremely high yields of MCFAs from organic waste. The bottleneck is actually more on the front end, converting those complex polymers to just lactate. That's actually one of the key challenges. Yeah.
We have a question online from Claire. I am working with a genetically intractable anaerobe, so I'm very interested in your methodology for methylating the plasmid for transformation.
Yep.
Could you expand on this approach, particularly the methylation sequencing you mentioned?
Yes, great question. And so hopefully we'll publish this pretty soon. And so the way it works in general, I don't have a detailed slide on this, but I'm happy to just chat about it. Okay, so the main challenge, right, you have a plasmid that you're trying to move into an anaerobe that has a system that recognizes foreign DNA based on its methylation pattern, right? And it's going to chew it up if it's not methylated correctly. And so what we do is we do, long read kind of the next gen sequencing platforms like PacBio and Nanopore. You can do methylome sequencing. And so when you do that methylome sequencing, what you can do is look at motifs that are corresponding to the methylation patterns. And then you can use informatic tools. One of the main tools is called RE-base to try to link that methylation pattern to a putative methylase in the genome of the organism. And so then that gives you a target to then try to express. So you've linked a methylase in the genome to a putative methylation pattern in the host. And so now you have the methylase. In our case, the one for Clavry was CKL2671. That's just one of the open reading frame locus tags. So now we take that locus and we express it in E. coli. And so we have we're doing this via conjugation. So in our conjugal donor system, Nigel Minton's group has done a ton of work on this. So go read his papers. He's a real genetic expert. We're just trying to work with him to get the tools set up. But you express that methylase in your conjugal donor strain such that prior to mobilizing the plasmid to your organism of interest, it gets methylated correctly, and then it can evade the host's or the organism's native RM system. That's the general gist of it. So another person who's done some nice work on this is Adam Gus at Oak Ridge. So I would say check out his work as well. Yeah, they're quite, Adam Gus and Nigel Minton, among other labs, are real genetic engineer pioneers for anaerobes especially. So check out their work.
Question online. I am very curious how you measured the carbon source utilization. Was this done in liquid culture with a plate reader and mass spec?
Yes. So how did we look at carbon source utilization? So there's two things. If we're looking at simple substrates, glucose, xylose, amino acids, etc., those things we can do using HPLC or GC, depending on what, classical analytical chemical techniques. If we're looking at waste, right, that complex mixture of waste, we can look at some key intermediates, like lactate concentration going away. But typically in our bioreactors, we look at a property called chemical oxygen demand. And so we can take our waste, which is this really complex mixture of organics, and we can chemically oxidize it, and then look at the amount of oxygen that's produced associated with that reaction and back calculate the strength, if you will, of the organic matter that's taken place. So we've done that. We've actually been very interested in just waste characterization as a whole, because in that mixed soup of pulp, like knowing exactly what the composition is very challenging. And so we've been just initiating a project trying to more precisely look at waste composition using what are called glycomic techniques, which can look at chemical linkages between different polysaccharide groups. And we've been looking at before and after degradation, what's going to be degraded, what's not going to be degraded. So yeah, so answer the question though, HPLC, GC for simple stuff, and then more complicated stuff is harder. But we can still do complete mass balances using COD.
So a much less technical question, since it's out of my area. But two thoughts came to mind. I mean, I know like Heifer International has this thing where they have a local thing with harvesting methane gas for cooking stoves.
Yep.
And it occurred to me, and also because of Diana work, that, I mean, you're dealing with a very complex waste stream that you're trying to deal with, but there might be really real opportunities for simple waste streams to do this very efficiently.
Absolutely, yes, that's a great point. So yeah, and that's how, so the cooking stove, right? That's what the biogas goes on to RNG, natural gas, it's cooking stove. But there's people working with acid whey, more readily degradable, other industrial waste products that have a very high strength, so they have to be treated, but are generally readily degradable. Coca-Cola's waste, for example. Sugary, really easy to degrade, and so very amenable to this approach.
Coconut shells is a real problem, for example. So I was just thinking that, I mean, it's really cool that you're doing this, but the applications to these, and it would be economical for the companies to have this, because then they would not, instead of having waste disposal, they would get another product.
Absolutely.
The other thing that is that it seems when you have a bioreactor, you get a lot of heat. Mm-hmm. So that's something else that could be captured? Is that something else that you're thinking about?
We're not looking at that. That is an interesting question. So the first, to award your first question, like that really speaks to like the tech, right now we're focusing on SSO because there's just a lot of it in society. We all generate it. So if we can have a technology that addresses SSO, again, thinking about global, like the widespreadness of the technology, we could really have it scale, and then that helps address the climate change issues, right? But it does have a wide mouth, so to speak. The second question, I just lost it. What was the second point? Heat, yeah. So we're not looking at heat recovery per se in this. So so biogas, people will take the biogas produced and make combined heat and power. Sometimes it's not as economical, or just use biogas directly to heat homes, things like this. Oftentimes in actually just wastewater, so in municipal wastewater systems, there's been some really interesting, innovative heat recovery projects actually. So I know that I'm from Vancouver, I spent a lot of time in Vancouver. In the Olympic Village there, they had a really innovative heat recovery project because wastewater comes out hot, right? And so they were recovering actually all the heat to heat all the homes and apartments in the Olympic Village during Vancouver. So certainly an opportunity. I think not directly connected to this, but in terms of resources to recover, yeah, heat's one of those things, right?
Just in Iceland, and in parts of Iceland, everything is geothermal.
Yeah.
And
Yep.
It seems that we generate an awful lot of waste that's some other way that we could think about recovering heat for heating homes, for example.
Yeah, a good point. In Toronto, actually, the university underwent one of the largest geothermal projects, I think, in the world, and a large chunk of the university is now heated all geothermally. So the big green grass at the university there's a massive geothermal infrastructure underneath that now to heat a lot of the university.
Other questions? Well, let's think. Oh, sure. Go ahead, Luis. We have plenty of time.
Sorry. Let me preface this by saying everything I know about biochemistry I learned in like the last hour. So I'm interested in the result that you saw, that the slower they grow, the more they produce these long medium chain fatty acids.
Mm-hmm.
I'm wondering if you've tried something where you just slow down the growth. I mean, thinking about biochemical reactions and like their kind of rates, and if you just have more time to sort of maybe, you know, stochastically go through these processes multiple times, I can imagine leading to more medium chain fatty acids, but just try slowing down the bacteria.
Yeah, and so you're dialing in on kind of, is there a selection parameter maybe we could use? And so you can control in the reactors, you can, in a continuous flow process, you can control the growth rate of the microbe using the retention time, how long it hangs out in the reactor for. And so that's a parameter you can push, make the SRTs kind of longer to maybe get MCFAs. Although in practice, separating out butyrate versus C6 production is challenging that way. And so controlling how much lactate you have versus acetate is an easier control. Yeah. But I mean, to this, right? So natively the microbe kind of, there's two strategies, right? The organism that's making C6 and C8, it seems to have evolved, right, in nature to be really efficient at lactate utilization, but again, at the compromise of having a slower growth, right? But its ecological niche is high lactate utilization. The organisms like in the gut of the cows and humans that largely are making butyrate, their ecological strategy is growing as fast as you can. And you can grow faster if you make just butyrate, but there has to be acetate around. And so that's kind of what the analysis is showing. Yeah.
Very thought-provoking talk, and we will resume at 11 a.m.